So this is our first speaker is Manel Esteller. So Manel Esteller graduated in medicine from the University of Barcelona, and he obtained also his PhD there in molecular genetics of endometrial carcinoma. And then he became the leader of the National Center of Oncology in Spain, so focusing on cancer epigenetics. And so he studied the alterations of DNA methylation, histone modifications, and chromatin in human cancer. Then he became the director of the Cancer Epigenetics and Biology program in Barcelona, and he's currently the director of the Josep Carreras Leukemia Research Institute, chairman of the genetics, chairman of genetics in the School of Medicine at the University of Barcelona, and ICREA research professor, so here in Barcelona. So his current research is devoted to the establishment of the epigenome and epitranscriptome maps of normal and transformed cells, and the development of new epigenetic drugs for cancer therapy, which is a highly complex scientific problem and technological problem too. So Manel, thank you very much. So the floor is yours.
There's no laser, no? Nothing like that. I can point, no? Like this. Okay, good morning, everyone. I would like to thank the organizers for their kind invitation to be here. When I received this VPH invitation, I thought it was HPV. VPH is virtual physiological human. HPV is human papillomavirus. Okay, so it's that way around. At the end, it's the same. So HPV, it's a good model to understand systems medicine. For example, what is better, vaccination or early screening? This is interesting because you can model that. What is better, to vaccinate everyone against HPV, or you have a good screening? And both are probably equally useful. So I'm not an in silico medicine person, but I'm inorganic medicine person. But I use a lot omics, multiomics, to understand biology and medicine. And this work is done now here in this building, white building in the other side of the city. I would like to start with this slide. At the slide that shows a problem that I like is what defines identity. What defines our identity as humans, as organs, as cells, as tissues. And this is a very complex, because there are different chemical levels of complexity that defines all these different biological samples. And in fact, all this can be modeled by different aspects. For example, we look in the morning, we can be old, but we look as if we were young still, like we're in our 20s, but we're not anymore in our 20s. In fact, what's interesting is that all our tissues, all our organs, they have the same DNA, absolutely the same sequence, but they have different phenotypes, different aspects. And this does not relate to the genetic sequence, that is the same, but relates to the regulation of the DNA and proteins that regulate DNA. So same genes, same DNA, but a different activation. What it means is same hardware, but different software at the end. This software of our cells is what we call epigenetics. It's over the genetics. And these are chemical marks that regulate the activity of DNA and proteins that regulate DNA. The most famous one is DNA methylation. DNA methylation is a mark, this one here, that DNA is A's, T's, C's, and G's, 6,000 millions. And the C can be, instead of a C, a cytosine can be a methylated C. And this usually is associated with silencing of the underlying sequence. Another level of complexity of epigenetics is that every DNA in our cells measures 2 meters. And it's not straight, because then we look like a star, okay, more or less. But it's highly compacted around a ball of a protein that we call histones. And this package is different in every cell of our body. And these are the histone modifications, the other component of epigenetics. When we have a stem cell, a cell that is able to generate any tissue, we're not changing the hardware of the cell. What we're changing is the software. We're changing the instructions that provide a differential gene expression. And this is what we talk again about epigenetics. The stem cell generate any cell from the brain, from the kidney, without touching DNA structure, just touching DNA regulation. The same when we have a cell from the skin, a keratinocyte, and we can use a stem cell. And in this case, thank you very much. And in this case, here it is. In this case, we have this cell from the skin, and we're able to generate a stem cell. And we're changing here epigenetics in the way around. And there is a third phenomena that we can have a cell, and we generate another cell without this intermediate stem cell. And we call this transdifferentiation. Transdifferentiation, it's a way to escape from therapies. Imagine that you have a prostate cancer. This prostate cancer is sensitive to a drug that inhibits hormones, the androgen. It's androgen-dependent prostate. So what happens is that these cells, they select a different program, that now this prostate cancer became a small cell neuroendocrine cancer of the prostate. And this tumor type is not sensitive to anti-androgens. So we changed the software to create a different cancer that now is resistant to the therapy. This is just an application of Darwinian laws at the level of cells and organs. So what we have shown is that these changes in transdifferentiation involve changes in DNA methylation. It makes sense, because this is the program that we use to express different genes at different tissues. And this is an assay here that it measures how different fragments of DNA that are very far away in a straight line in the 3D space of a cell, they're extremely close. Okay, like in the wormholes in the galaxies that can be very close, this is the same. In the DNA, sequences that are very far away, many thousands of nucleotides away, in fact, in reality, they are extremely close because like a fold paper, they touch each other. Okay, and in the leukemia, they do not touch, but when we transdifferentiate to another cell, they are touching each other with a change in DNA methylation. If we go back to my second slide, identity, why we are different in this room? Kind of different, because we have some difference in our genetic sequence, because we have difference in our copy number, of how many genes we have. But we are different because we have a little bit different software programs that regulates our expression of genes. And this was the first study that in humans, looking at 3 human populations, only looking at DNA methylation, we're able to say, this sample has to come from somebody from Han Chinese American, for example, only looking at the DNA methylation sample. And this has to be African American, and this sample has to be Caucasian American. Only looking at the software, not looking at the DNA straight in the DNA. Some of these variants are irrelevant, almost irrelevant for pathology, but some of them are involved in disease. And for example, we identify that one of these variants was associated with an increased risk of developing colorectal cancer, that one of the most frequent tumors that we have. An important difference between genetics and epigenetics is that epigenetics is pretty static. More or less, we die with a similar DNA that we were born, but in our lives, our epigenetic configuration changes a lot. And it's very dynamic, and we can change it also with drugs. These are drugs that we can give to the cells that they change the epigenetic landscape. These drugs are inhibitors of DNA methyltransferases, of histone deacetylases, etc. There are many drugs. This is a big market now in pharma. They are developing these drugs that interfere with the epigenetic setting of cells. But what's important at the end of the day is that some of these drugs has reached clinical stage, and those that are here with this highlighted here, these are drugs that they're approved for clinical use in patients. They are not science fiction, they are science reality. They are drugs used in the treatment of leukemias, lymphomas, and sarcomas. So this was a great advance for the field of epigenetics. Maybe one of the most famous parts of epigenetics in disease is that in cancer, one way to inactivate genes that are anti-cancer is to silence these genes, to inactivate these genes. And they are not inactivated by a mutation, by a defect in the hardware, but they are only inactivated by a defect in the programming, by silencing these particular genes. This is promoter hypermethylation. And these are genes in all the hallmarks of cancer. For example, this is a gene here that we call cadherin-1. Cadherin-1 is a gene that is like a glue that fixes together normal cells. Okay? What happened in breast cancer? That this gene is silenced because it's not expressed, you don't have the glue, and the breast cells, they dissociate to each other, and this contributes to the generation of metastases. Because metastases are cells that are not fixed together. They're able to escape in the bloodstream and generate a metastasis far away. The changes in DNA methylation are not only in promoters, as I mentioned, but they can be changes in distant regions of the genome, as I described before in the example of transdifferentiation. I want to tell you something that probably you may know, is that our DNA, only 10% of our genome produce messenger RNA and proteins. Only 10% of our genome produce proteins. Proteins is what we see each other. We see our proteins and a little bit of fat, mostly. Okay? This is what we see each other when in the mirror and in the metro. Okay? But only 10% of our genome produce proteins. There is a lot of DNA that we call dark genome. That genome is not a DNA sequence from aliens. This dark genome there is really some of the sequences that are producing what is called non-coding RNAs. Non-coding RNAs are ribonucleic molecules that do other things. They do not produce proteins. They do other things, like regulating messenger RNAs, for example. And these are non-coding RNAs with different functions that we have. Some of them, you have never heard of them. So who cares about this? No? So what are these relevant? These guys here, for example, tRNAs are very small. These are like a message of nothing. These are 17, 18 base pairs. This is nothing. But if we don't have these guys, there are no testes in the wall. There are no testicles in the wall. Okay? So at least for half of the population can be important. Okay? We have here another interesting one, is someplace, schist, so it's here, schist. Schist is another non-coding RNA that you may think is not relevant at all. It's more bigger, it's around 500 base pairs. And schist is the molecule that allows the existence of female in the wall, because it's the molecule that inactivates the X chromosome in females. In females, you have two X chromosomes, and one of these should be inactivated, okay? Because if not, you will have a lot of problems, let's say that, and you will not be fertile, etc., etc. So females are females because they have this schist gene there, okay? It's not a gene, does not produce a protein, it produces a non-coding RNA. What we have found in these last years is that many of these components are altering cancer. In cancer, you see defects in all these non-coding RNAs, epigenetic defects that change the expression and activity of many of these molecules. And in recent years, if you thought that this was complicated, the picture is now more complicated, because RNA itself can also be modified. We don't call it this epigenetics, we call it epitranscriptomics. Everything that regulates the molecule of RNA. And this is here. This is a complicated slide. I'm not going to explain it, but DNA can be methylated. So RNA can be methylated like DNA, but at the C, at the A, the U of RNA can be a pseudo-uridine, etc., etc. Many modifications. And what we have seen this is just work from my lab, but this is done also by other labs, is that many of the proteins that take care of the epitranscriptome, they're altering cancer, and they alter many of the activities of RNA molecule. Okay, so this is like white knowledge, an introduction to this complicated field of epigenetics, epitranscriptomics. One application of this field is, for example, in cancer, to try to translate these discoveries to improve a more target therapy of cancer. In the cancer warrior, to identify what is the weakest point, what is the defect that we can hit without killing all normal cells. And for example, this field started many years ago when we discovered that in brain tumors, gliomas, when you have silencing of this DNA repair enzyme, these patients are sensitive to drugs that damage DNA. And now in the hospitals, when you have a brain tumor, they analyze this gene, and if you are positive for this methylation, they will give you a drug. And if you are negative, they will give you another treatment. Okay? It's a way to classify and to give the better treatment to these patients. This is another example in breast cancer. This is another DNA repair enzyme, BRCA1, and in this case, also the methylation predicts response. Here is what we call a comet assaye. Does not relate to astrophysics, but what relates is the formation of this tail. It means damage of DNA. And these cells, they undergo damage of DNA, and then they die because they have methylation of this enzyme, and they cannot repair DNA damage. So it's a way to classify breast cancer according to this marker to say these patients should receive, in this case, what is called a PARP inhibitor. The world is not perfect, as you may already know, and some cancers, most cancers, if you use only one agent, they develop resistance. They survive, they adapt, they use a Darwinian lifestyle to undergo survival. And for example, we can put a tumor in a mice here, and the tumor that is sensitive to this drug is sensitive because it's methylated BRCA1, and you kill 90% of the cells, but 10% reappear, and these 10% that reappear after the treatment, they have changed the epigenetic setting, and they are, for example, unmethylated. They express the gene, and they are now resistant to the drug. So the drug changes the setting, the programming of these cells. So epigenetics and epigenomics participates in sensitivity, but also creates resistance to the drug. In this case, to the shield, to the arrows in this warrior. The next slide is the only nasty slide I have, just to let you know, because I saw it here. And it's this one here, so to make you awake. And this is a slide that shows also the reality sometimes. And this is a patient that have melanomas. Melanoma is a tumor from the skin. It's not the common one. It's from the nevus here. And these melanomas, they have a mutation in BRAF, and they receive a BRAF inhibitor. And you see how clean they are later. At 15 weeks, the tumor is gone. But at 23 weeks, the tumor reappears. And the tumor reappears in the same place. This tumor here is this tumor here. This tumor here is this tumor here. It's in the same place, but the tumor is different. The demographics of that tumor is different. It has a different software. It has a different expression of thousands of genes. It has adapted to survive in the context of the drug. And of course, epigenetics play a role of that. And this is an example where we use cells of colon cancer were sensitive to this drug and cells that were resistant. And among these, there were no genetic difference. The only difference was one gene in more than 30,000 that had a DNA methylation difference between this and this. And this is this particular gene that is involved in the repair of damage. I want to give you some examples of further examples of real-life studies in the lab to help oncology patients. And this is one study that I also like because it shows this idea that in a tumor, not all cells are created equally, and they do not develop equally. So there are difference amongst cancer cells in a given tumor, in a patient. And in this case, we just took in a colorectal cancer, colon cancer, and we took three parts of the tumor, this part here, close to the light, in the middle, or close to our body here, and analyzed DNA methylation, using DNA methylation arrays that analyze more than 1 million sites in the genome. And we saw difference between these regions. So there are intratumoral heterogeneity. This intratumoral heterogeneity is one of the problems in oncology. It's what generates chemoresistance in human tumors. Another interesting study, I would say, that has rich clinical application that we did in the lab is this one here. And this is a study that really involves the use of artificial intelligence and other technologies, computational technologies here. And this is a study that tried to address the issue of cancer of a non-primary. This is a question in oncology of around 2.5%, 5%, no more of cases, that these are patients that appear a metastasis, but the medical doctor doesn't know the origin of the tumor, doesn't know where it came from. So this is disseminated disease, and you don't know what it is. So these patients have a very poor outcome. So what we did here is we took a lot of this cancer of a non-primary, we took a picture of them. This is a genomics picture that analyzes, in fact, DNA methylation sites. It's an epigenomic picture of these cases. Red, it means methylated, green, unmethylated, different 1 million sites there. And we studied a cohort of tumors that we have, all tumors. Imagine here there are more, close to 3,000 human tumors. And we obtained the profiles of DNA methylation of each tumor. And then, when we had these profiles that define each tumor type, we throw our problematic cases of cancer of a non-primary. And then, using the model of a discovery cohort and validation cohort, we're able to say, in each case, what was the origin of CAP, because it had a profile that was similar to one tumor type or the other. There are profiles of DNA methylation in CAPs that they look alike a pancreatic cancer, they look alike a colorectal cancer, they look alike lung cancer. Okay? This is very similar when you are in a small town, you live in a city, you go to a small town for holidays in the summer, and you are walking around and say, where are you at? You are from that family, no? Because they recognize your face. This is the same. It's the same story. You have cancer on a non-primary, we take the picture, and this picture look alike a colon cancer, pancreatic cancer, breast cancer. So we can give a diagnostic to the medical doctor, and he can take then the medical decisions there. Another use of epigenomics is to decide, for example, what is the use or not of immunotherapy in some cases. Immunotherapy is a wonderful new treatment in cancer, but only 1 in 3 patients respond to immunotherapy, more or less. So what happened in the rest? So we obtained, using this way to study, using a discovery cohort and a validation cohort, we analyze hundreds of tumors, those that respond to immunotherapy, those that do not respond, and we obtain a bioinformatics signature that we call epimmune, that if you are epimmune positive, it means you are going to respond to therapy. These are typical Kaplan-Meier curves that is a way to display survival of patients. The patients that survive are higher, and those that die sooner, they have this type of curves. An epimmune positive is a patient that responds to the therapy. There are other markers that you can add there. For example, a patient that's epimmune positive, PD-L1 positive, CD8 high, a lot of mutations, a lot of antigens in means here. So this is a great responder to immunotherapy, the best responder. But if you are epimmune negative, PD-L1 negative, CD8 negative, low mutational load, immunotherapy is a waste of money, a waste of time, and you can create further damage in that patient. So it's important, this type of biomarkers, to define the right therapy for these patients. I want to introduce briefly to you another technology and therapy that is the most new therapy in cancer that we have. Instead of giving compounds to the patient, let's give it cells to the patient, the right cells. And this was patient 0. Patient 0, like in a zombie movie, is the first patient that had the disease. And this case was the first patient that had the treatment. She was Emily Whitehead. She was here extremely young, as you can see here. She had a B acute lymphoblastic leukemia, a B leukemia. All treatments has failed. And in this picture, she just had a few weeks to live, 3-4 weeks, and she's going to die, according to statistics. She was saying goodbye to her puppy here, okay? And she received a therapy with T cells that were able to eat the B cells of the leukemia. This is what we call CAR T cell therapy, CAR T cells. These CAR T cells, at the end, they are T cells you take from a patient, you have them in the lab, you make them really mad. It means you play with them, with the genetics of these T cells, and you show the picture of a B lymphocyte, particularly CD19. It's an antigen of that lymphocyte. And then when they're really mad and they know their enemy, you put them back in the body of the person. And these cells run like crazy by all the blood looking for B cells to kill them. Okay? And this is the basis of CAR T cell therapy. And you have this patient, this person, more than 10 years later, this beautiful girl here, that she's completely cured. And that's amazing because I said this was 3-4 weeks, very close to die. I know this is a therapy that is given in the world, but it has problems. Problems, very expensive. So we have to decide how to do these treatments. So imagine that one patient, it cost 250K euros, okay? So it's a lot of money. So it will be nice because it's expensive. There are sometimes adverse effects, and sometimes patients do not respond to therapy, to have a way to select what is the right patient to receive this drug. So what we did here is we did a clinical trial, putting together our patients with patients in Israel and in Italy. And these are CAR T cells used for B leukemia and B lymphoma. And we obtained the epigenomics profile of the cells just 1 minute before giving to the patient. The cells just before being used in these real patients. And from this, we obtain a signature that we call EpiCard. This EpiCard signature that came from the bioinformatics study, again, using the discovery validation pipeline. If you are EpiCard positive, it means that these CAR T cells are extremely good. These are cells that are going to do their job. But if you're EpiCard negative, these cells are not so very good. They're kind of lazy cells. These are T cells that will not do a good job killing the leukemia cells. And this relates to the cell types that are there. I'm going to go in details. And now this is a test that you can use in these patients. But not only that, now there are clinical trials that they mix immunotherapy plus epigenetic drugs. The epigenetic drugs awake immunology of the cells. They allow the expression of antigens. And the immunotherapy then can act better in those patients. So it's a good combination of drugs. So as you can see, so my lab does a lot of studies in human cancer, analyzing different markers that can be single genes or can be epigenomic signatures that are derived from biocomputational studies of our cases. And this together allows a better personalized therapy in cancer. Of course, I fully recognize that complex diseases like cancer, they require the efforts of many experts. And if we think that cancer like an elephant, we only look at epigenetics, where maybe this guy here, just looking at the tail. I'll say, how is an elephant? I will say, it's a tail. It's very thin and very long. But if we call this guy here, that can be a genomics person looking at copy number variation, say, how is an elephant? And it's long, but it's kind of white, okay? And we told this guy here, what's an elephant? And this can be, the bioinformatics guy, say, bioinformatics, well, it's extremely hard because he's talking here this particular part of the elephant. Only putting them together, all these experts, you have a complete picture of the elephant. Not the elephant in the house, the elephant that is the problem we're trying to address. And for example, studies in this way that we have put our expertise with expertise of other people. This is a study that we did with the scientists in UK from the Sanger Institute, where they had, we analyzed 2,000 cancer cell lines where they had the treatment against 300 compounds. We added epigenetics, now transcriptomics, metabolomics, genomics there. And this data is available to everyone. So you can go there and say, okay, what about this gene, this drug, and expression? Any relation between sensitivity to this drug? And you can do this analysis. And in fact, now, many articles in the world, they start using this database first. And later they have their own data and validation, experimental validation. But we like it to use a lot of this data. This is free to everyone. And this is an example of collaboration between different labs. Another recent example of collaboration is this one that I like it really a lot also. In cancer, we have sometimes these weird patients that we call exceptional responders. This is around less than 1% of the patients. These are patients that they have an advanced disease, and according to statistics, they are left only, again, 1 month to die, more or less, according to statistics. You give the usual drug, okay, I expect nothing, really, and these patients, they have amazing response. They have amazing response, the tumor disappear. How is possible? What they have so special? These patients, in fact, they have been also called Lazarus, like in the Bible. So they are close to be dead and they are alive. How is possible? So what we did is put together a task effort between different experts, and we analyze in detail all the multiomics of these exceptional responders in the world. And what we realize is that they have something special. And what's special about them is that they have defects in the same pathway that is targeted by the drug. Let me explain you. One example, glioblastoma, GBM, is one of the worst tumors you can have in the history of humans. But this patient, after receiving the classical drug, it's called temozolomide, she was alive more than 5 years, while she should be dead at 8 months, more or less, according to statistics. So what's going on? How is possible? What this patient had, she had 2 mutations in DNA repair enzymes and 1 methylation in another DNA repair enzyme. So in this case, when this tumor was hit by a drug that damaged DNA, could not escape. The cells die because cannot use any metabolic, another pathway to escape, because all of them had these defects in the same pathway. This is unusual, because according to Darwinian laws, you try to have mutations at different places, not accumulated in the same place. So that's the reason they are so unusual. But now there is a clinical trial trying to generate mutations in the same pathways and to create exceptional responder patients. And this, I would say, is very interesting. We have learned a lot from this study. I want to mention briefly multiomics beyond cancer. And this relates to all human diseases. For example, rare disorders. Rare disorders, many times, this happens in kids, these rare disorders, many times there is a mutation, germline mutation, in an enzyme that we call epigenetic enzyme. And the phenotype is usually mental impairment or a defect in immunology. Among these, the most famous is probably Rett syndrome. Rett syndrome, it's a defect that they have mutations in MCP2. This is a protein that binds to methylated DNA. These girls, the boys, do not survive a pregnancy. They die like embryos and fetus there. These girls, they have a defect in MCP2. Because they have this defect in this enzyme, they do not care about the programming. They do not care about DNA methylation because they are missing the protein that recognizes the signal. And you can see here, this is an assay to measure that. This is this patient with this mutation. You don't see any band here because the protein cannot bind to DNA, this mutant protein. So they do not care about the software because they are not able to distinguish between methylated or unmethylated DNA. They're not able to distinguish between an on-off signal in the DNA. Analyzing the genome of these people and the expression, we have seen that they have defects in inflammation, for example, because they have this defect in MCP2. And in this case, we have used therapies with an inhibitor of inflammation that really diminish the clinics of these animals with the defect. This is not curative therapy. The only to cure these people, you can only have to use gene editing and introduce the right gene. Take out the mutant and introduce the wild type. Okay? But this so far is not possible. Remember, this is in the brain, so it's very difficult to do that. Talking about the brain is, for me, is the most interesting tissue, and it's the only organ that studies itself. And in humans, in other species, but let's talk about humans for now, it's a very dynamic organ that changed the software epigenome a lot. From 0 to 15 years old, there is a lot of changes in the epigenome programming. Okay? From 16 to 64, there is like a plateau. There is some increase, but a plateau, and later there is a decrease. What is this period of time, from 0 to 16? This is the period of our lives that we learn a lot. We learn most of what we are. Okay? We learn how to talk, how to eat, how to go to the bathroom, how to relate with each other, etc. So the bad news to all of you is that after 17, you don't learn so much. Okay? You just learn a little bit in the university, it looks like that. And after 64, you start losing stuff. Okay? And you can lose very slow, or you can lose very fast. And very fast is neurodegeneration, Alzheimer's disease, for example. One point here, this is learning, a learning time lapse here, but when you reach 16, 17, this is a time that sometimes appears a disease that can be schizophrenia, or can be bipolar disorder. This is the mania and depression here. And we think that these are really diseases of the software. There are no mutations, there are no genetic defects. This is at the last point when your brain is almost perfectly wired, something goes wrong. And the brain doesn't get the right expression of genes, okay? The right way to express genes. And these are different defects in DNA methylation that we have observed in the brain of these patients. If you look at the end of the process, after 64, started neurodegeneration, you see that the frontal cortex of Alzheimer's has defects in DNA methylation, for example, in the way we process beta-amyloid protein. The same is true not only in the cortex, but inside, in the hippocampus here. These are defects here in the hippocampus. In this case, this affect the metabolic pathway of the tau protein that relates to neurodegeneration too. And it's interesting that many of the dementia associated with all this neurodegeneration, if you look, these are complete epigenomes. The same way you have a complete genome, you have a complete epigenome in this case, that all these dementias, some share common traits, common genes affected by changes in the programming. So there is something that is similar to all dementia. It doesn't matter if it's an Alzheimer, it's a dementia with Lewy bodies, a Parkinson, or a dementia in Down syndrome. They share something. These hallmarks of dementia, of neurodegeneration. The same that there are hallmarks of cancer, there are hallmarks of neurodegeneration. And some of them relates to these changes in epigenetics. And you can mix both of them. For example, these are families. You are born with a defect in what is called an enhancer. An enhancer is a region of the genome that regulates another region of the genome, but very far away. Only in 3D they get together, okay? And these people are born with this mutation enhancer. The enhancer cannot bind to the gene, and the gene became methylated, and this gene is silenced, it's not expressed. And this is associated with higher risk of developing Alzheimer's in the lifetime of the people that have these defects here. Of course, epigenetics is not only cancer, it's not only rare disease, it's not only neurodisorders, it's also metabolic disorders like diabetes type 2. Are also cardiovascular disorders here. This is an aorta from a patient with a cardiovascular defect that has can have mutations in lipoproteins, can have high fiat, but have silencing of genes that protects against the damage of the aorta at the same time. So there is, all diseases in human are multi-complex, multi-omics, it's involved in all of them. I would like to to switch to more weird stuff a little bit. I think they have more weird stuff. No, not so weird. So I told you that there are changes in the brain that happen in aging, but we wanted to assess changes overall in aging, and we studied the epigenome of people like this guy here, newborn babies, and people like this other guy here that they are over 90. As you can see, the phenotype is very different between them. The phenotype is pretty different. But the genotype is almost the same. There are very few mutations difference between this guy and this guy. So everything is software, a wrong software of these people. And in fact, this is the case. When we analyze, this is a way to represent the epigenome of these people. This is the newborn baby. This is a person of 50 years, and this is a person over 90. We go from the dark blue to the light blue, and this is not like the wine, the tasting you're going to do on Thursday or so. This is not like the wine. It's worse in this case. It means that as we age, we lose the right software. And for example, we have hypomethylation. This relates to chromosomal instability. And we have hypermethylation of tumor suppressor genes. And this relates to higher risk of cancer. There are a special type of humans that they have what we call accelerated aging. And these can be patients with premature aging disorders like Hutchinson-Gilford progeria and Werner syndrome. And these are kids. These are kids that they have 8 years, 9 years, 10 years, no more. But if you look at them, they look like 80, 90, 100 years. How is possible? These people, they have these phenotypes because they have a mutation in DNA repair enzymes, so they cannot repair DNA. When we do a test of DNA methylation of these people, our profile of epigenome is telling us that these people, they have 80 years, 90 years. They are not 8 and 9. Biologically, they are 80 and 90. And this is an important point, the difference between chronological age and biological age. Chronological age is the day you are born until now. Biological age is how life has shaped your cells. And this can be different. And in this case, these people, they have a chronological age of 8, biological age of 80. That's the reason they die when they are 10. Because when they are 13, they are 130, 140. Okay, so the reason they die when they are teens. So a way to measure age now of our cells, biological age, is using epigenome. They are called epigenomic clocks, that at the end are not so different than the way to measure age in the trees. Like our father once decades ago brought us to the woods to show us the age of trees. It's not so different, the type of representation that we do. I would like to mention close to the end that this knowledge of epigenomics can be applied to any disease, even emerging disorders. And in this case, we apply this to COVID-19. COVID-19 has been a very bad disease in the sense that have killed millions of people around the world. But most people, they were pretty resistant to the disease. They did not show a very dramatic phenotype. So what is the difference then? Why some people get infected and have a very serious disease with respiratory defects and others are completely fine? So what we did is we took a picture. Again, this is the epigenome picture of people that they have a bad disease and had a disease that was asymptomatic or was treated just at home. And we identify a signature, the epi-COVID signature, that shows that the people with the disease that require, that was associated with respiratory failure, they have a particular pathways affected by this disease. These pathways, at the end, what are telling you is that people with COVID-19, that they had a respiratory failure, they had an over-response of their immune system. It was not itself the virus killing the lung cells, it was our own immune system killing our lung cells. There was a response that was too much in this case. This was true in this COVID in adults, but was also true in COVID in kids. Kids are these amazing creatures that they can be very resistant to the COVID-19. Okay? They do not realize, they continue playing football or whatever they are doing. They do not stop. But from time to time, we have kids that upon infection with the virus, they develop this syndrome that is called MISC, multisystem inflammatory syndrome in children, that is different because it's not so lung. It's not in the lung like in the adults, but affects the heart, the skin, the kidney, etc. And this can be a very, very bad disease. So what's going on with these kids? And these kids, again, when we obtain an epigenomic signature that predicts if you are going to be a MISC kid, somebody with a predisposition to develop this adverse reaction to the virus. And again, we use most of the time this pipeline of a discovery cohort and Bayesian cohorts comparing people with the disease and without the disease. And here, at the end, these kids with the disorder, they have an overactivation of the immune system. Again, too much immune response. That's the reason they have this very bad disease. Just to mention very briefly that this was MISC, but in Asia, in 2009, 14 years ago, there was an infection by this type of virus that induced a high number of Kawasaki disorder disease, and these kids looks very similar to the COVID-19 MISC children. And we apply our signature, epigenomic signature, to samples from these kids. And in this case, all of them had the same signature. So I think that we have identified a signature that is not specific of COVID. It's a signature that predicts that if you are a kid and you receive any virus, you're going to have an excess immune response. And then you can have Kawasaki, MISC, and other disorders. So I think this is a signature of overreaction to virus in kids. So this is an example of translating one signature from one disorder to another. And I would like to finish, and now it's true, about this study that we just finished recently, that I still think that science should be driven by curiosity, by applications, of course, but by curiosity. So picking here in the future, so I'm going back to the past. In the past, we show that there are special type of humans that we call monozygotic twins. Twins are clonal. It means that they cannot be differentiated according to genetic sequence because they are clone of each other. They have the same DNA sequence, okay? But some of them are more fat, more slim, more shy, more brave, okay? Shorter or taller. And this does not relate to the genetic sequence because DNA is the same. And we discovered years ago that this was related to the epigenome, to the software, different software they can have. And in this case, these twins, for example, they have this gene that in one of them was methylated and the other was not. And this was the difference between them. And this started a field that now there are hundreds of papers looking at discordant twins. One twin has ELA and the other doesn't. One twin has cancer and the other does not. One twin has diabetes and the other does not. And they're looking for epigenetic difference between these discordant twins. And there are hundreds of papers of that. But so we decided to look now something a little bit related to this. And trying to answer this question a little bit about nature versus nurture. Not to answer, to help answering this long-standing question there. So we started to decide to study people that look alike, okay? So this, we call them like VPH, we call it virtual twins also, because these are virtual twins, or look alike humans. People that they look the same, or very the same, but they are not related. They are not family. They are not twins. How is possible that? How is this convergent phenotype of these people? And this is the study here that we have done, and you can check. So what we did in this study is to put together lookalike humans around the world, okay? Trying to collect samples from these lookalike humans around the world. And this is the distribution. These are humans around the world. And these people here, they are not twins. These are people, they look alike. They look very similar, but they are not related in any way. You can go back generations, there's no commonality. They don't know each other, but they look very similar. Some of them can open the iPhone of the other. So how is possible? How these people look the same? they look alike. So first we decided, okay, are they really look alike or not? So what we did is we ran through them different software recognition programs. These are the same programs that are used by police to select if you have been against or pro or whatever thing in the streets. Okay, the same they use in police, the same used to open when your face to go through the planes. Now these are the same type of programs. Okay, and at least half of them were almost indistinguishable, were the same according to the 3 software programs. So they were not able to make them apart. They were extremely similar. So we studied carefully these lookalike that were very similar, and we did a multiomics study. Studied genomics, DNA sequence, epigenomics, DNA methylation, and the microbiome, the composition of bacteria and virus that they had, to see what was providing these similar phenotypes. And this was the study. And the study at the end shows that what put them together, what clustered them together, was genetics. They had more common genetic variants between them. They share genetic variants. And the difference were associated with DNA methylation and bacteria and virus. But what put them together was really genetics. They share these genetic variants. So despite they are not related, we are so many humans in the world that by random chance, we're starting to produce humans with similar genetic sequences. And this is a very interesting concept there. And the young kids now, they can go online and find their partner, lookalike partner. And there are different apps to do that, okay? So these people are put together by DNA variants. What are these genes? These genes are genes that relate to bone formation, genes relate to the skin type, genes relate to cell junction, and genes relate to ion binding. Remember, we have the bias that we selected these couples for the face. So our genes relate to the face, mostly. So it makes sense. Bone, the skin type, cell junctions, how tight is your face or how puffy it is. And here the same, ion binding, because it's the way how we retain water. You have a more thin face, or you have really this round ball moon face, okay? How you retain water there. So this provides the shape of our face. This, just one little note, although they were selected by having the same face, we realized that they have other things that they look alike. For example, to use the right hand or left hand, so both usually are right-handed or both are left-handed. One is a smoker, the other is a smoker, and vice versa. Okay? And this relates to addiction. Addiction is highly inherited. The way we are addicted to things, there are people that have addictive personalities and people that they don't have it. Okay? This is a lot of the genetics there. So they look alike beyond the faces. But this is a discussion for other people to do research because it's highly complicated how to address those issues, okay? But really, our face is telling more than really facial features. Anyway, and the final consequence of these studies are several. One is that now, from the DNA, for straight DNA, we can start building faces, okay? And there are different programs now in development, some of them already being used by police, trying to construct the face. And it looks that they work better than the witness. The witness in a crime is not so good as the program of the DNA. Now you can reconstruct better the face using this, but we can also think about the other way around. And what about if from a face, now you can draw the genome of a person? You can dissect the genome, obtain the genome without sequencing. Only the face will tell you the genome. This can have interesting uses. For example, in genetic disorders, just looking at the face, you can say, according to this face, maybe there is a mutation in the receptor of this neuroprotein. Okay? Something that we can try to think. And this can be extremely useful. Just to let you know that this is not science fiction, and in some hospitals in the US, when you go in, they take you a picture and they say, okay, my advice is go to cardiology, go to neurology. Okay? So it's a way just to do some pre-screening, just looking at the face, because the face is telling you important clues about your genetic sequence. So I would like to finish here to highlight the relevance of the genome, but also the relevance of what's below the epigenome. And I would like to thank all the members of my group and all our supporters. Thank you very much.
Thank you very much, Manel, for this brilliant and enlightening talk. So it is open for question. Yes, so here in the back, Bart is giving you the mic.
Thank you very much for this talk. It was very, very interesting and also very easy to follow. I have a question. I understood that there is some epigenetic hallmarks of diseases, and every disease, every type of disease has its own epigenetic hallmarks. But how much is known about how epigenetic then influences at the biological level the expression, for instance, of the gene? Yeah. We have a lot of information, or a field of study.
Yeah. So this is a gradient. It will be easier. This like is an on-off mechanism, but it's not. So there are some epigenetic marks when they go together. So it means that that gene is not expressed at all, 0%. But some of these codes allows 70%, 50%, 40%, 20% expression, etc. So there is a degree. It depends on the combination. For example, you have DNA methylation in a highly dense in a promoter, proximal promoter, and you have histone modification marks, specific ones. This usually is associated with silencing in more than 90% of the cases. If they are not there, it is possible that express, but you require transcription factors to go in there. So I say that it's a good marker of silencing of the genes, and not so good of how expressed is that.
Okay. And another thing, it was like the last study was super interesting. How many repositories did you check for finding similar people around the world? Because
so we had to get ours. There's no repository. So the first ours was the first repository of these people. So we're able to get around 64 people so far. It's very difficult because and we did this before it was popular. Now we'll do it another way. Now I'll use Insta or something like that. Okay, because now there are apps that the teenagers, they're looking for their twin, and they want to date them. Okay, it's very interesting, all these things. Yeah. Okay, it's a different world. But if I started this, I'll do it a different way. But we started this study like 6 years ago or more, or 8 years ago. Yeah.
Very, very nice. Thank you.
Okay, more questions? Yeah. What?
Thank you very much for the talk. And I think it's going to be a little Black Mirror question. So do you think there's going to be, for example, like in the future that you're going to have schools that say, okay, genetics influence your IQ? Or like you go and you find a lawyer and you're saying, I'm very violent. Is it my genetics? Yeah. At what point can you say, is it me or is it my genetics? Can you just blame everything on genetics?
Yeah. So if we... the most complicated part of phenotypes is behavior of humans. Behavior of humans is one of the more complex ones. Behavior of humans involve intelligence, as you mentioned, that is not intelligence, it's intelligences, they are different, and behavior of violence. For example, there's only very few cases that you have inherited violence cases. And there is a family that all the families, the grandparent, the father, the son, all of them were highly abusive, beating their wives, killing people. So how is possible? All these people, they inherit a mutation in the androgen receptor, a highly dominant mutation. Okay, so they were overactivated for this. This is rare. This is rare. We cannot blame we cannot use this kind of excuse. But there are families, very few families that have this. No? So there's some genetics that is very strong. But in most of the cases, you have this regulation, and this regulation of the penetrance of the trait. And this penetrance of the traits regulates to all these markers that I mentioned today: DNA methylation, histone modifications, etc. This is the same like inherited cancer. Inherited cancer is only 5–10% of the cancers. Okay, is what you have this mutation, you are pushed to have the cancer. But even in these cases, you can regulate that. For example, you have a high risk of breast cancer, that your diet can modulate the risk to low down the risk. If you have kids earlier or not, modulate the risk. The age of the first menstruation, all this regulates the risk. So yes, there are some genetics that is very strong. It's not the usual cases, but there are some. But in many cases, what we do is able to modulate the risk.
First of all, I want to thank you so much for this interesting talk. Just wanted to ask, so from this, is it possible for later to have like a sort of reverse mapping of the genome based on a picture or like just from like a video call in order to predict a disease or predict a disorder that might come later in life?
Yes, so let's say now when we are born, we can do a genetic sequence, and from this you obtain a probability of developing disorders. If you add, when you are born, some of these changes will provide you even a better probability. But all these are probabilities. What you do in your life is very critical to finally develop the disease or not. At one point of your life, the genetics is the same, but you can deduce from this some of your risk of cancer or other disorders. For example, if we do a test, if you are 28 years old, but your DNA methylation clock is telling you that you are 45, it means there is something wrong there. So, and it's something that is very interesting, and many people are doing that, are doing this test of biological age in the same way that they are doing tests to know where I am from. I'm from Israel, from Italy, from North Africa. Okay, so now you can do a test that can tell you, okay, you are 25, but you are not 25. You are 45, according to your biological age. The good news is that this can be modified. For example, people that are smokers, they age faster. We know that. When you stop smoking, you're able to regress a little bit in your age. Not maybe to catch up fully, but do improve, and many other examples.
Okay. Thank you.
Hi, I'm Charles McKenna. Thank you for the lecture. It was actually very interesting. So you mentioned trying to induce group genetic mutations for DNA repair for those super patients that respond well to the regular drug therapies, but what are the side effects of that?
Yeah, so this is a clinical trial that's going in the US, and this is done in a very controlled manner. So it is done only in the tumor area, so it's not mutations everywhere, so it's not they can inherit, etc., and it's done in that area. Remember, these patients, they are in a desperate situation, so they are going to die in 3-4 months. So in this case, when they wait one and the other, so let's do it. Let's try to do it in a clinical trial. We can induce sometimes particular mutations. It means let's do a change in G2A only. Okay, we are able to do that because we know with this change creates sensitivity to this drug. Okay, it's not only in the gene. We have different genes that are mutated. Some are good, but in that one too. So it's done in a very controlled manner in the tumor and only for the type of mutation. Not the gene, we're not so good on that, but in the type of mutation.
Okay, thank you for that. Also, one other question. You just mentioned how people that smoke have increased aging.
Yeah.
Would the same happen for people that vape?
What?
Vaping?
The vegan.
For vaping e-cigarettes.
Oh yes, and we have not done the study. There's no study out there. Again, it will not be surprising. It will not be surprising. People that age very fast also are chronic carriers of infection, HIV or E. coli. These are also have a premature aging. But from those that are vaping, I will not be surprised.
Okay, thank you very much.
So I do have a question, maybe, if I can.
I think here's one in front.
Is there
Fatima?
a question there?
From front.
Fatima. Go ahead.
Thank you very much. It has been extremely interesting. So I have two questions. The first one is that you have been explaining us the importance of epigenetics, but what gives these epigenetic abnormalities? So because it has been said that it's mainly environment, so it's our diet, their pollution. So what can we do to improve our epigenetic? And then if you can comment a little bit about prenatal life, that you have skip it in one slide, you have said, okay, prenatal life is But there is a lot of epigenetic changes in this part, no, in our prenatal life.
Yeah. So we have an epigenome that defines us like a species. Homo sapiens has an epigenome that is different than Neanderthalensis and others. Okay, so this is what our basics are. But we have variation between humans. Okay, species, variation between humans, and then we have variation in disease. And this variation in disease, some of this can be inherited if you have this particular human variation, and some of this can be acquired by environmental changes. The number one environmental change that causes downstream effects is tobacco, smoking. This is number one. Second is radiation, high-dose radiation. The third probably are now small particles from cars and others. And the fourth one will be food. Mainly for methyl donors and acetyl donors. Okay, because if we take a lot of methyl donors, this came from the folates, we'll have too much methylation. But if we take too low, you have too little methylation. The same for acetyl-CoA is this, because this is acetylation of histones. Okay, so these are the factors that really determine in the environment all that. About the prenatal exposure, it is extremely relevant, and we have the case of many kids that they don't have any genetic defect, but they have important problems in school. Because they have, for example, alcohol exposition in prenatal. So this alcohol exposition that we see now in a lot of adopted children, because we don't know the mother what we did, it was usually an alcoholic. This alcohol, it changes a lot the profiles of mostly histone modifications. So it's one factor that is very relevant there.
Thank you very much for this very interesting talk. I want to generalize it a little bit because, you know, the public here is predominantly engineers. And nowadays, well, this is the VPH summer school. So one of the things is simulating things. This is what we want to do, where you want to start from some type of knowledge, whether it's biochemistry, whatever, and you try to see whether you can replicate that and see what involves in this. So this is really trying to understand the different processes. And you've shown a couple of things there also, where generally you go from trying to find a pathway or whatever, and then say, okay, we can explain this, and we can find this, and we can use this. On the other hand, what we see nowadays more and more is that we have machine learning. So you take a huge amount of data, and you see whether something comes out. You do some clustering, for example, in order to find things. And some of the examples you showed is where you have all these kind of new biomarkers from epigenetics that show, okay, your response to cancer is much better than the others. But when you look at your curves, still only half survives after 6 months or a year. So how do you explain that then? Is this data fishing? Would you have the same results when you would work under using DNA, for example, or if you would work on, I don't know, school results or something like that? Or do you really think that this approach learns us a lot besides statistics? And how do you think both should interact?
So the same type of representations are for genetics, for expression, for everything. There's no much difference. Remember, these are complicated cases. So we can do the same from non-skin melanoma cancer, and you'll see 90% survival. Okay, that's the reason I don't study the tumor, because if they survive 90%, it's not my problem anymore. Okay, so I prefer those tough cases, okay, that I have more problems there. So the same is for all. But yes, as you mentioned, we're a lot of interested in the prediction, have a model to predict, without even the real sample. And now some of these models can predict the behavior of samples that we have not seen. And this is what is relevant really about what I like of this type of summer school. Just to mention, my lab now has 4 people that they are not at all biology. They have not seen a cell in their lives, okay, a cell or a tissue. And these are people that are mathematicians. And we have engineers so far, but it's mathematicians and others that in informatics mainly. So these are essential now. Because as you said, it's not fishing before. Now you have this information, I want to study this gene. Now you like to obtain global patterns, an interaction between patterns, an interaction between all the multiomics that you have there. And what is more relevant for the other? What determines one versus the other? These kind of things. And for this, we require all these experts.
But when you look at this, then what do you think is most important? Is that this kind of data analytics, so the type of machine learning and then trying to find patterns in there, or the one that are trying to understand the physiology and from there going? And how should they interact?
Yeah, yes. So this is, so as you know, the old school are the second one and the new school are the first one. And this is at least for me, are equally important. I'm not a partisan in this, because some people say, no, only studying from this, from the physiology, I can understand this. And I say, no, this is old-fashioned. We have to look globally, and then we'll find this. I think that both are equally important in this case, because I'm only driven by the results. What is the final result of this? Is something meaningful one way or the other or not? Because sometimes starting for a protein that is mutated in the domain, blah, 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 at the end there's nothing. I see nothing. And vice versa. I see people that are putting together 1 million samples, and I don't see any conclusion. What is the conclusion of these 1 million samples you put together? There's no conclusion. So I think that, but in other cases, both are able to obtain meaningful results. So I will say that here is that, what is the meaningful result? And it doesn't matter if it comes from a single case carefully studied, okay, or it came from many, many cases that you apply all these tools. I think that at the end, what's important is what is the final result of this. And later you can cross it, of course, all the times, because now you have this particular gene, protein, sequence, you can apply it in a large data set, and vice versa. Your global results can be applied in a real patient or not. In one person has any value, that or not. So I think that's the Ocan cut at the end, okay, the result.
And with regard to research, so you say you have these biomarkers, and as I mentioned, like 50% predictive in some ways. But the other 50% are not predictive, so that means that you can't use the epigenetics, or at least not that epigenetic signature. How do you proceed with that? What should be done in order to go further and study this kind of population?
Yeah, so it's not 50%. Sometimes it's 5%, sometimes it's 70%. 50% can be the average. It depends. It depends on the tumor, or depends on the disease, and all that. Some cases, for example, in immunotherapy, it's only 30%. And correlates pretty well with the real-life scenario that only 1 in 3 cases respond to immunotherapy. What is the response? The response is the multiomics. So that's the reason I like this study, is that you mix everything. You mix epigenomics, RNA, genomics, copy number variation, microbiome, of course, exposome, everything that is around these cells. Because you have more information, you are less likely to fail. Really, and them are complementary. I think that the exceptional responder is a good example of that. The exceptional responder without epigenomics will not be there, but without genomics will not be there. So it's only when you have these patients that have this particular setting, touching different genes by different ways, genomics and epigenomics, they say this is an exceptional responder. So only mixing together all these multiomics, you have the answer.
But that, of course, comes then to the problem in getting your data, because if you need multiomics, you need good data of all the omics. How do you do that in reality?
Yeah, so there are now tools that, first, sequencing is going cheaper. And now you can do sequencing in the same sequencing. You can analyze DNA methylation also in one shot. Before you have to do 2 different techniques. You can do it in the same shot. You can do RNA expression. So you're putting together in one shot, one type of technology that does 3 things together, and the price is going down. So I think eventually you'll be able to look at this in one shot.
Thank you.
Okay. So I do have a couple of questions. My first question is a bit related to this discussion. You've shown a lot of examples where you can very nicely explain post hoc what is happening in terms of mechanistically, but I'm wondering whether you can do it forward so easily. Up to which point the effects, the epigenetics and the effect thereof is stochastic or really can be deterministic. I was struck, for example, by the example of the melanoma, no? Where the cancer rose again, so with different programming. So I was thinking about this adaptation of bacteria to a specific treatment that then escaped the control of people. So would you comment on that? So is there specific patterns so that we can think to understand one day, and in order to use that in a predictive way for drug design, for stratification?
Yeah. So I think that there are different types of answers to your question, but let me give you one example. When we have a cancer in a person, and you take a tumor that sometimes can be pretty big, it can be like this, okay? In that tumor in a person, you have a lot of defects at the same time, many. And the natural history of those defects can be very different one person from the other. So imagine that you have a person with a colon cancer with a mutation in a gene that we call p53. This mutation, say, okay, this is the driver or whatever, the mutation. But what caused the mutation p53? So we know that now there is an epigenetic silencing of a gene that creates that mutation in p53. So, okay, then the driver is p53, so this silencing. Okay, what caused the epigenetic silencing of the gene? So we know there is a mutation in another gene that induces more methylation of this gene. Okay, so this mutation in the gene generates this mutation. Okay, but what caused the mutation in the gene? We know that hypomethylation of the genome, hypo, creates a chromosomal instability that breaks down that gene. Okay, but what caused hypo? So we can. This discussion can go forever and ever. Okay. Well, the reality is that in that patient we have all this. Okay, and we can trace back, and people are doing that. They're tracing back, doing trajectories, and now we're doing the same that I explained today at single cell level, because this is bulk. I said one example, but we're doing the same using single cell technology that requires a lot of computational power, etc. And we're getting this back, and we're seeing that many patients have different trajectories. This is individual, and we never know what is the first cause of any of these diseases. It's very difficult to know, because even in families with a mutation, well, the mutation is there. What happened with that mutation? And there are different penetrance of the disease. This is one type of question. The other question is, most of those studies in biology and medicine are retrospective, so at one point in time. Okay, the prospective studies are more difficult to do for many reasons. For example, because sometimes they are never published because they require 5-10 years later of the study, and who cares 10 years later about what happened 10 years before? The field has moved forward from that. So there are very few prospective studies of anything in medicine right now, but there are a few. For example, in our detection of cancer of unknown primary, this was retrospective, but it's used in the clinics, for example. And now we know that if this patient, they receive the right treatment according to the tumor, they live longer than if they didn't know anything about this tumor. So this is prospective, but it's very difficult to do a prospective study in medicine right now. Most of them are retrospective. And later they came prospective. That usually are cases, real patients that go there.
Right, so that's nice because it comes naturally to my second question, is about the determination of baseline. So you've talked about atherosclerosis, you've talked about disorders that are not obligatorily cancers, and so the problem here, so what was first, no? The chicken or the egg? So I guess there is a very strong difficulty then to establish a baseline, because there are as many baseline as persons, probably. So I guess there is a lot of values of the longitudinal data that you can extract from population cohort. I've seen that you're using Twins UK that has longitudinal data. So how active is the use of this cohort in the field of epigenetics?
Yes, so longitudinal data, it is great for us because it can show you how things change. No? For example, we're just finished a discovery about in one type of leukemia, how use of an epigenetic drug in the longitudinal manner change the course of the disease. And we have different time points, and you can see how that is done. So yes, these are critical samples, longitudinal samples. They're a bit difficult to get, the samples. And to obtain the baseline, so even the genetics, they don't know that baseline. Now there is a big achievement to have a pangenome. What are the pangenome? In fact, there are a few samples. Because until now, all the genomes were of male, white male. And this was our reference for more than 25 years. And now we have put a few African-American samples and a couple of Asians, and this is the pangenome. No, it's not the pangenome. It's an approach to the pangenome. Okay? Even for genetics, that should be easier. They don't have a reference baseline. So we have some kind of a reference baseline that we use. So imagine for the other marks, for microbiome, for epigenome, for transcription even. So it's very difficult that. So we have to compromise. This is for our population, this is our baseline. Okay? And that's it. Because when we have a disease, it's clear the disease. It's more clear what's a disease. No? Cardiovascular, you see it. Almost with the naked eye, you see this is a tumor. This is an aorta that's obliterated with fat. You can see it there. But the baseline is more complicated. And what is the baseline? Taking in consideration the population here in Catalonia is over 60. So the baseline should be 60 years, not 24. So what is the baseline?
Great, thank you. So I don't know if we have some questions online.
So if you're online and you have a question, please raise your hand and you can connect to the microphone or put it in the chat, one of the two. In the meantime, any other questions here in the room?
So maybe I just have a curiosity about regulation that is not biological regulation, but more low regulation. So then you've showed that example that sounds a bit futuristic, but you're saying it's not that futuristic because in the US it's already being exploited, which is then the link of between facial recognition and epigenetics hallmarks, and so probably then the fate of the patients. So in Europe, there is very strong, regulation about the use of data, especially those data that allow identifying the person, identifying the hallmarks of the family of that person, etc. But now, if you start to go that deep, in the biological curriculum of a person, of a family, just looking at the face. So, are you aware about the regulatory challenges it poses?
Yes, I think it poses the same regulatory challenges, not the same, but close to other tests. For example, I remember perfectly well when the first genes of inherited breast cancer were discovered and how insurance was trying to take benefit of that. So that will not give you an insurance if you are BRCA1 carrier. Of course, this discrimination that cannot be applied. They cannot be applied, at least in Europe, these kind of things. But for years, you can do a blood test and tell you what is your levels of your fat, of your cholesterol in your blood. And this can be discriminatory. Because if you have high cholesterol, you're going to live less. Okay? So why I have to take, you have more disease? Why I don't want you to be a customer of my insurance company. I don't want to hire you because you're going to have a lot of problems. This has been protected. Okay? So the same has to be with all these new technologies and this all new knowledge. They has to be protected in the same way that it was protected blood analysis, germline mutation, DNA recombination, etc. It requires the same level of protection. That is the toughest that we can. Because the medical applications are going to be extremely good, but the bad applications can be also terrible. So we need the same tough regulation that we have done for all the other tests.
Okay, thanks. Then I guess that defining the levels of clearance for specific groups
but
is
it will be the same that for genetic disorders, and for example, this law against discrimination. You had a cancer in the past, you should have the same chance to get a job that you were cancer-free. Okay, these kind of things, they should be absolutely pushed forward and should be. It doesn't make any sense, but still people trying to discriminate. You had a cancer 12 years ago. That according to medical oncology, you have 5 years you are cured. So it doesn't matter you were 12 years ago a cancer.
Okay, great.
Maybe just a final question. You say that you have in your lab engineers, scientists, mathematicians, and so what do you find important in their profile? Should they know something about biology, about medicine, or should they only know about machine learning or about statistics?
So let's say two things. One, it would be nice if you know something about biology. But since a lot of people know about biology, so it's something. It's an easy field that engineering or biology is very easy, at least for me. So it's very easy. And the second, open mind. Open mind because they have to collaborate and co-work with people that doesn't know anything about engineering. Okay, so engineers they know something about biology. Biologists they know very little. They know how to do R, and that's it. Okay, some of them, the good ones. Okay, so that's the end of the story. Some very small programs, some Python. So it's almost nothing there. Okay, so open mind because you have to collaborate with that people that they don't know anything about this. So I think that collaboration is essential. And we have, for example, joint programs ourselves with the Barcelona Supercomputing Center. They're going to visit, and we have co-students together. Have joint programs, and they are mixed, and this is great. This is great for all of us.
Okay, thank you.
Very good. So thank you again, Manel.
Thank you. Thank you.